Hi everyone, welcome back to A Level Biology Help. Today I'm going to take you through the photosynthesis section for AQA A Level Biology. Also, at the end of the video I'll be going through a few exam questions and explaining their mark schemes. And as always, I'll be putting timestamps in the comments section so that you can skip to different parts in the video if you do not wish to watch the whole thing. Right, so let's get started. So these are the content that I'm going to go over today. So first I'm going to take you through the light-dependent reaction, the light-independent reaction or the Calvin cycle, and then I'm finally going to go on to the limiting factors of photosynthesis. So let's get started with the light-dependent reaction. The light-dependent reaction, as the name suggests, relies on light. So it uses light energy and water to form ATP and a molecule called NADPH, which stands for nicotinamide adenine dinucleotide phosphate. The light-dependent reaction occurs in the thylakoid membrane of the chloroplasts. If you're not familiar with the parts of the chloroplast or the chloroplast as a whole, please go check out my video on the structure of eukaryotic and prokaryotic cells. So the first step in the light-dependent reaction, or the first stage, is the photoionisation of chlorophyll and the photolysis of water. Then we have the chemiosmosis process, or the electron transport chain, to produce ATP. And then we have NADPH production. So first I'm going to take you through the photoionisation of chlorophyll. So here we have a pretty terrible drawing created by me of the thylakoid membrane. So we have the thylakoid membrane here, and then we have the stroma, which is like the space inside the chloroplast, and then the thylakoid space. And if I just get my pen tool out, I'll just label the different parts. So these big green structures here, which are pretty terribly drawn, are called photosystems. This one here is called photosystem 2, so just write PS2 for short. And this one is called PS1. Now a lot of students tend to get mixed up between which one is PS2 and which one is PS1, because they normally tend to think that this one is PS1, as that is the one that comes first in the many diagrams, but it's the other way around to what you may think. These red-like structures here are called electron transport proteins, so I'll just write ETP to make things easier. And then this funky looking structure here is an enzyme called ATP synthase, which produces the ATP from ADP and PI. And then this dark green blob here in PS2, and there'll also be one in PS1, is the chlorophyll molecule, which gives the plant its green colour. So chlorophyll contains electrons, and when light energy hits the thylakoid membrane, it passes through photosystem 2. The energy from the light, which comes from the sun, excites the electron, so it gives it more energy. This means the electron rises to a higher energy level. This means that the chlorophyll molecule that the electrons were in has become photoionized or oxidized as it has lost an electron. Now these electrons are replaced by the process of photolysis. Photolysis is the splitting of water using light. So the light hits the water molecule and splits it into oxygen, protons and electrons. The electrons are then reabsorbed by the chlorophyll molecule. The importance of the protons will become more important when we talk about chemiosmosis. So what is chemiosmosis and the electron transport chain, or the ETC? Well, once the electron that has been excited to a higher energy level has reached the higher energy level, it is transported through a series of electron transport proteins down an electron transport chain. As this happens, the electron loses energy or releases energy. This means that protons, or H+ ions in the stroma, can be pumped through to the thylakoid space. So it gives them energy to be pumped through like this. This creates a proton gradient in the thylakoid space, or an electrochemical gradient, we sometimes call it. This generates what we call a proton motive force. This means that protons can then be pumped through the ATP synthase enzyme. This causes this blue part of the enzyme here to rotate, to cause ADP and inorganic phosphate, or PI, to combine to make ATP. Now, with these diagrams, I've demonstrated this using just one proton, but I think it's about 6 protons are required to produce a molecule of ATP. And obviously more electrons would be used, not just one. So NADPH, or nicotinamide adenine dinucleotide phosphate, is produced. So the electrons that have lost energy from the electron transport chain appear here at the bottom in the thylakoid space, and they appear near photosystem 1. Then light strikes photosystem 1 and the electrons are then excited up photosystem 1. These directly reduce NADP plus to NADPH, or we sometimes call it reduced NADP. So that is the light-dependent reaction, as it relies on light to produce NADPH and ATP. So now we are going to talk about the light-independent reaction, or the Calvin cycle, which is named after the scientist who discovered it, Melvin Calvin. So the light-independent reaction occurs in the stroma, which is the space in the chloroplast. Again, if you're not familiar with that, please check out my video on the structure of eukaryotic and prokaryotic cells in the corner. So the first step is we have the carbon dioxide, which is produced from the Krebs cycle, which we will talk about in my next video. And we have a molecule called ribulose bisphosphate. Ribulose bisphosphate is a 5-carbon molecule. So the carbon dioxide and the ribulose bisphosphate combine to form a 6-carbon molecule, but then they split, which is catalyzed by the enzyme Rubisco. This forms 2 molecules of a substance called glycerate 3-phosphate. So 2 3-carbon molecules called glycerate 3-phosphate. The glycerate 3-phosphate molecules are then reduced by NADPH with the help of ATP to provide energy. So it's a reduction reaction to produce 2 molecules of triose phosphate. Now these triose phosphate molecules can either be recycled to produce ribulose bisphosphate, hence the name Calvin cycle, or they can be used to produce other useful organic substances, for example polysaccharides, e.g. cellulose and starch. If you haven't watched my video on carbohydrates and polysaccharide, please check that out in the corner. Or they can be used to be converted into glycerol, which combines with fatty acids to form lipids. I also have a video on lipids if you want to watch that video. So there are limiting factors of photosynthesis, which are factors that affect the rate of photosynthesis. The first limiting factor is temperature, as this affects the rate of reaction and enzymatic activity. Then we have carbon dioxide concentration, which affects the rate of the light-independent reaction. So it affects the reaction between carbon dioxide and ribulose bisphosphate in the light-independent reaction. And the other main limiting factor is light intensity, which affects the rate of the light-dependent reaction, as the light-dependent reaction relies on light to excite electrons to a higher energy level, and therefore their ability to be transported down the electron transport chain to produce ATP and NADPH. Now, agricultural practices can overcome these factors, so reduce the effect of the limiting factors. One way in that this can happen is through the use of LED lights to grow plants under. This maximizes light intensity whilst maintaining the temperature, as natural light affects the temperature quite often. So here we have an image of plants being grown under LED lights. Next thing is that you can use a greenhouse. This controls the light intensity and the temperature to maintain a high rate of photosynthesis. Also, you can burn fuels to produce more carbon dioxide. Now you can see the advantages for this as it produces a high rate of the light-independent reaction. However, you may be thinking that this will produce a greenhouse effect as carbon dioxide is produced, so it is bad for the environment. Right, so that is it for the content, and now we are going to go through a few exam-style questions. So I'll just get my highlighter out. So, chloroplasts contain chlorophyll A and chlorophyll B. Scientists found tobacco plants with a mutation that caused them to make more chlorophyll B, so we can highlight more chlorophyll B, than normal tobacco plants. So the production of more chlorophyll B is caused by a mutation. They investigated the effect of this mutation on the rate of photosynthesis. So they are investigating the rate of photosynthesis, so we can highlight that. The scientists carried out the following investigation. They grew normal and mutant tobacco plants. They grew some of each in low light intensity and grew others in high light intensity. They isolated samples of chloroplasts from mature plants of these both types. Finally, they measured oxygen production by the chloroplasts they had isolated from the plants. The figure below shows the scientists' results. So here we have on the x-axis the light intensity in micromoles of photons per second per meter squared, and then we have the oxygen produced in micromoles per milligram per hour. And here we have this top line here, which is mutant plants in high light. Then we have normal plants in high light, mutant plants in low light, and normal plants in low light. So let's look at the first part of the question. Explain why the scientists measured the rate of production of oxygen in this investigation. Now, as this is an explained question, you have to justify your answer. So you have to explain why something is happening. Well, your first thoughts may be that they're measuring the rate of production as oxygen is produced in the light-dependent reaction. So this will give the scientists some indication as to what the rate of photosynthesis is. So this is what I've written. Oxygen is produced in the light-dependent reaction through the photolysis of water. This means that the faster the rate of oxygen production, the faster the light-dependent reaction is. So let's look at the mark scheme. So the first marking point says oxygen is produced in a light-dependent reaction, so we will get that mark. Then this question says the faster the oxygen is produced, the faster the light-dependent reaction. So we would get both marks for that question. So let's look at the next part. In each trial, the scientists collected oxygen for 15 minutes. Calculate the difference in the oxygen produced by the chloroplasts from mutant plants growing in low and high light intensities at a light intensity of 500 micromoles of photons per meter per second. Show you're working. So this is a nice maths question. So I need to get my pen tool out here. So the first thing we need to do is to look at the oxygen produced in mutant plants in low and high light intensities at 500 micromoles. So first we need to look at the x-axis and read to where 500 micromole is. So this is about here. So if we read up to first the mutant plants growing in low light, this is about here, and we read along, this is here. Now each square on this graph is quite hard to see, but it means 10 micromoles per milligram per hour. So we can say that this value here is 60. And then if we read up again and look at the mutant plants growing in high light, it's quite easy to read, it's 200. So low light we have 60 and high light we have 200. So we need to find the difference in the oxygen produced. So therefore we need to subtract 60 from 200 to get 140. Now you're probably thinking, oh, we've got our answer, it's all fine. However, the units here say micromoles of oxygen per milligram per hour. However, the scientists only collected the oxygen for 15 minutes. This means that we need to divide this by 4. as there are 4 15 minutes in an hour. So 4 times 15 minutes in an hour, so 140 divided by 4 equals 35. So our answer would be a difference of 35 micromoles of oxygen per milligram per hour. So let's look at the mark scheme. So the mark scheme allows you to write any number between 35 and 36 as the graph. The measurements of the graph are sometimes subjective. So 35 to 36 micromoles of oxygen per milligram of chlorophyll. So we would get both marks for getting that correct answer. However, it says here you can get a mark if you have the correct difference at 500 micromoles of photons per meter per second, or incorrect difference but division by 4. So if you got any of those points correct but you got the final answer wrong, you can get 1 mark. So let's look at the last part of this particular question. So here we have a copy of the graph and the text. So the question here says, the scientists suggested that mutant plants producing more chlorophyll B would grow faster than normal plants in all light intensities. Explain how these data support this suggestion. So remember from the question that more chlorophyll B is produced when the plants are mutant. So we need to look at the mutant plant data. So as you can see, the mutant plants give a higher rate of oxygen production, both in the high light and low light. And we need to explain our data, so we need to say why this happens, not just what the pattern is. So this is what I've written. So I've written, at all light intensities, so at high and low light, chloroplasts and mutant plants have a faster production of ATP and NADPH. So you need to refer to the products in your answer as the question is referring to growth. So as you can see, they have a faster rate of production of these molecules, as the gradients in the mutant plants are higher than that for the normal plants. So I've also put so there is a faster light-independent reaction. So more sugars, for example glucose, can be used in respiration to produce more energy or more ATP for growth. So I've referred to the uses of these products in growth. So let's look at the mark scheme. So at all light intensities, chloroplasts and mutant plants have faster production of ATP and reduced NADP or NADPH. Do you get that mark? So they have a faster / more light-independent reaction. So they produce more sugars that could be used in respiration to have more energy for growth. So you'd get all 4 marks for the question. Now another point that you could have also put is they have a faster or more synthesis of new organic materials. If you put all 5 of these points correctly, you don't get any extra marks, as the mark scheme says 4 maximum. Also here it says accept converse points if clear answer relates to non-mutant plants. So you need to refer very clearly to the non-mutant plants if you want to get any marks if you wrote the converse points. Right, so let's get on to the last question. So a student investigated the effect of different wavelengths, so this is what they're investigating, of light on the rate of photosynthesis. So this is what they are measuring, so this is a dependent variable. She used the apparatus shown in figure 1. So here we have this nice piece of apparatus, which is a kind of round bottle-shaped apparatus here, with a suspension of photosynthesizing algal cells, an oxygen meter to measure oxygen production, and light of a particular wavelength being shone on the cells. So the first part of the question says, what measurements should the student have taken to determine the rate of photosynthesis? So as you can see by the apparatus, we have an oxygen meter. So this suggests that the student is using the rate of oxygen production to measure the rate of photosynthesis. Now, especially from GCSE, you should be able to recall what the rate means. So if you apply this to the question, we can say oxygen production, so that is what you are measuring, so that is the independent variable, and time. So you need to put both of those to get a mark. So the mark scheme says oxygen production or oxygen concentration, it doesn't matter which one of those you put, and time. So as the word time is underlined, you need to mention both to get the 1 mark. so we would get that 1 mark. So it accepts oxygen volume or concentration. However, it rejects oxygen uptake. It rejects oxygen uptake as oxygen uptake is to do with respiration or aerobic respiration. Also, neutral reference to carbon dioxide uptake, as they are measuring the light-dependent reaction and carbon dioxide is part of the light-independent reaction. So if we go on to the next question, other than temperature and pH, give 2 factors which should be kept constant during this investigation. So you just have to name 2 other limiting factors of photosynthesis, which, as you may be able to recall from earlier in the video, are carbon dioxide concentration and light intensity. So a pretty easy question. So if we look at the mark scheme, you can write the intensity of light or light intensity. It also accepts distance from light. You can also put the amount / number / mass / species of algae / photosynthesizing cells. You can also put the carbon dioxide concentration or partial pressure, so we would get both marks. And you can put time also. Now, as the mark scheme says 2 max, if you put more than 2 of these points, so if you put 3 or 4 of these points, you don't get any more than 2 marks. Right, so let's look at this part of the question. So the student did not use a buffer to maintain the pH of the solution. Explain what would happen to the pH of the solution during this investigation. So it's asking you if the pH would increase, decrease, or stay the same, and why it would be like that, as it is an explain question. So this is what I've written. I've written that the pH would increase as more carbon dioxide is removed for photosynthesis. So more carbon dioxide being removed increases the pH. So let's look at the mark scheme. pH increases, you get that mark. Neutral becomes more alkaline or less acidic, so the examiner doesn't really want you to write that, it wants you to clearly refer to the pH. And the second marking point says as more carbon dioxide is removed for photosynthesis. So you don't actually need to write for photosynthesis or more to get the second mark as they are in brackets. So we would get both marks for this question. So let's look at the final part of the question for the video. So figure 2 shows the student's results from the investigation. So on the x-axis we have the wavelength of light in nanometers, and on the y-axis we have the rate of photosynthesis in arbitrary units. So the question says, suggest and explain why the rate of photosynthesis was low between 525, so we can underline that, and 575 nanometers wavelength of light. So as this question says suggest and explain, you need to come up with your own ideas from your own knowledge and also explain why you have suggested your suggestion. So the first approach we need to take to this question is to look at the rate of photosynthesis between 525 and 575. So 525 is around here, and 575 is around here. So as you can see, the rate of photosynthesis is quite low at this point. So you need to explain and suggest why it is so. So as they are measuring the effect of the wavelength, you can say that there is less absorption at these wavelengths of light, because if the chloroplast doesn't absorb the wavelengths of light, then obviously the excitation of electrons and photolysis of water cannot occur. Or you can say that the wavelengths are reflected more. And obviously light is required for the light-dependent reaction. So if we look at the mark scheme, so less absorption or more reflection of these wavelengths of light, so we would get that mark. Here it says reject no absorption or cannot absorb unless in the context of green light. So there is less absorption, but there's not no absorption at all. So if you put these points here, you wouldn't get any marks at all for the question, even if you got mark point 2 or mark point 3 right, because it says reject. Also, it says here that note, no green light absorbed or green light reflected, you'll get both marks for the question. So if you specifically refer to green light, then you'll get both marks for the question, so you wouldn't need to write anything else. So the second marking point said the light was required for the light-dependent reaction or photolysis. So we would get both marks for this question. Or you can put represents green light or the colour of chlorophyll. But if you wrote all three of these points correctly, you don't get 3 marks, you can only get a maximum of 2 marks, as the marking says 2 max. Right, that is all I want to say for this video. Thank you very much for watching. If you have any questions at all, please comment them below in the comments section. I'll be more than happy to answer them, and I'll see you in the next video.